we study the process through which single-celled organisms evolve into multicellular organisms. We're doing a long-term directed evolution experiment. I hope to run it for 50,000 generations or so before I die or retire. We're on generation 9,000 now.
Do you think that this could be what you're creating in the lab is actually a phase shift in evolution, and you could get something that's just a bit crazy?
It's possible.
Dr. Will Ratcliff is an evolutionary biologist running one of the most ambitious experiments in history, pushing single-celled yeast through 50,000 generations to recreate the evolution of complex life in real time in a lab. His groundbreaking work fuses synthetic genetics, computer modeling, and evolution to unlock mysteries of how multicellular life arose on Earth. Remember to subscribe to the channel to support the podcast. It really helps us out. And if you do, you will discover exactly what happens to octopus when they take LSD.
I think the origin of life is, if not one of the biggest scientific unknowns. Multicellularity has evolved not once or twice, but many times. More than 50 independent transitions to multicellularity across the tree of life.
Wow. Would you say that there is life on any planets that are relatively close to Earth, but we just don't have the chemical tests to determine that outright? I'm going to pin you to a yes or no on if you think we'll find life on other planets in our lifetime. When you're in a bar or walking down the street and someone asks you what you do for work, what exactly do you tell them?
So I tell them I'm an evolutionary biologist, and they look at me sort of confused, like, what is that? And, oh, I'm a professor. And they go, oh, what do you teach? And I say, I teach a class on the origin of complex life. I teach an intro to evolution class for undergraduates, also new graduate students. Sometimes I teach a class in sort of foundations of mathematical modeling and the quantitative biosciences. But teaching is like I consider a relatively small proportion of my effort. Most of my effort goes to research. And then that kicks off a whole conversation. Like, oh, what do you do in terms of research?
How do you explain that research? Because it's really quite a niche area. So how do you describe that research in simple language?
Totally. Great question. So I'm usually like, multicellular organisms are organisms like me or a tree or that dog, things you can see with the naked eye generally, right? And this kind of organism has evolved numerous times on Earth from single-celled ancestors, things which look like bacteria. And it's a key step, one of the most important steps in how life goes from simple beginnings to a more complex state. And so in our lab, we study the process through which single-celled organisms evolve into multicellular organisms. And we kind of know, broadly speaking, that this occurs through, the formation of simple groups, those groups becoming evolutionary units so that they reproduce, they have heritable variation, they're multicellular traits. Natural selection acts on that, and that process drives sort of the input of information and the ability for them to evolve into organisms. So functional integration and new traits at the multicellular level. But it's been really hard to study how that process occurs in nature because that happened hundreds of millions of years ago in the lineages that exist. And so in the lab, we're doing a long-term directed evolution experiment. I hope to run it for 50,000 generations or so before I die or retire. We're on generation 9,000 now. And it's just an open-ended, like evolution experiment, watching something go from single cell to simple multicellular state to increasingly complex. And we can, we're learning the process, even if we're not necessarily explaining how it occurred in the different lineages that have happened on Earth. We're learning a lot about the dynamics in the process through which multicellularity can arise.
So cool. It's so fascinating. Do you have any idea what the organism will look like at time step 50,000 generations? Which is just a mind-boggling sentence, first off. But, what was the organism in generation 1? Yeah. How is it different in generation 9,000? And then what's your intuition on what it might look like in generation 50,000?
So I'll tell you the truth, which is that I don't know what it would look like in generation 50,000 because I couldn't have predicted the first 5,000 generations.
Right.
And this is I think has been one of the more surprising things for me, that we understand evolutionary biology very well. We understand physics very well. And it's the interplay of these two things that are really driving the sort of future directions of our experimental system. And yet, even if you understand the sort of rules through which things update, you don't necessarily understand all the trade-offs that go into actually shaping how natural selection unfolds, and all the times that they can surprisingly break trade-offs that you think are ironclad. They work their way around them with really clever, innovative solutions. And so it makes it really difficult to predict evolutionary futures. And I think this is a fundamental thing in evolutionary biology. Some things are fairly predictable, just based upon, there are certain ecologies, certain ways of making a living that sort of pull biological systems onto manifolds, right? So you have similar, there's this whole field of island biogeography. When, when Victorian era naturalists started exploring the world, they were like, wow, in Australia, you have things that look like wolves, but they share essentially no evolutionary relatedness to actual canine wolves. They're, marsupial wolves. And that's because there's an ecology for wolf-like predators in many ecosystems. And so, and they have mice and they have, shrews. They have all these things which independently, convergently evolved traits that mirror what we have in Europe and the Americas, because there's an ecology that favors that. You have powered flight evolving in insects, in mammals, in reptiles, convergently, right? Because again, there's an ecology for it. So some things are sort of predictable. That kind of coarse level of prediction is like, you can sort of figure out where these manifolds lie and think that, organisms will be entrained upon that, because that's a kind of unbeatable strategy, right? The shark body plan hasn't changed.
Yeah. Sorry to interject. Could you explain quickly what you mean by a manifold?
I'm using it as a sort of a metaphor here, but like, if you think about the traits that organisms have in more than 3 dimensions, right? Like,
yes,
ecology is non-continuous, right? You have things that sort of look similar, and then there's a gap, and then there's things with another similar grouping of traits. This isn't just for plants and animals. This is true for microbes. This is true for bacteria. It's true for viruses. And to the extent that concepts like species explain the world, and this has been a very long debate in evolutionary biology, like, are species real? Are species useful? It's not as settled as you might think it is. I'm personally in the camp that, yes. And it comes back to sorry, I know I'm getting way off topic here, but this is fun.
Please.
It comes down to like a fundamental thing in biology, that biological systems are fundamentally continuous. And we, like, as humans, we like to discretize them and say, like, we had one species, it evolved into 2. At some point, it became 2 separate species, right? But like, okay, what is that point? When did you cross the threshold from 1 species to 2? And you could have like a very high bar, like there's no more genetic information being exchanged between them. But that type of bar doesn't really work for most things in the real world that we would say are different species.
Right.
It would be fine for me in an oak tree, but it wouldn't be fine for two different, very distinct oak tree species, right? And so that's an example of, like, there is eventually some point at which they're unambiguously different species, but like our intuitive understanding of what species are, things which have which kind of exist in these different high dimensional spaces of having traits and are discrete from one another, right? That you do see that pattern throughout the different sort of like across the tree of life. You see it for bacteria, right? You see it for viruses, and I think this comes down to the fact that there are certain ways of making a living that make sense, and you tend to have populations adapting towards that sort of manifold of what makes sense to make a living, right? And that gives you some differentiation, some discreteness between one population or species and another one, right? And you don't just have a big blur of everything being totally continuous.
Right. Makes a lot of sense. I think you're absolutely right that we humans like to categorize and label. So we put things into discrete buckets, but it's interesting to think about evolution on this continuous plane of traits, niches, behaviors, all of these different things. What would be some of those? What would be a concrete example that you've seen in your lab of something evolving that you really didn't expect to?
Sure. So let me tell a little bit more about our system, since we
Please.
haven't really talked about the sort of basic premise here. So about 15 years ago, I was avoiding like writing up my thesis as a graduate student. I was a fifth year PhD student, and I thought, like, wouldn't it be fun to try to really get at some of the biggest questions in evolutionary biology, like the origin of life or the origin of complex life. And I was talking to Mike Travisano, who had recently joined University of Minnesota, where I was a graduate student. And, we were like, we just don't know enough chemistry to deal with origin of life. Like, we're biologists, we're not chemists, which is still, I think, a very legit challenge I have with origin of life research. I love it, but it's too chemical for my knowledge
Right.
base. Like, what about multicellularity? And so we started playing around with evolving multicellularity in single-celled yeast. Yeast is a single-celled fungus, and we chose it for pragmatic reasons, that it's something which we know how to manipulate really well at a genetic level, at a culture-based level. It's easy to grow. There's a huge ecosystem of tools that you can do really cool synthetic biology with it, which opens up lots of opportunities for hypothesis testing if you can kind of add or delete genes, or even just custom order them and have them sent to you. Then you stick them in there. And we do that all the time in my lab. Like, we wanted to make our yeast use light for energy. And so Tony Bernetti, a research scientist in my lab, figured out how to take yeast, which to our knowledge are in a lineage that's never used light for energy in 3.5 billion years. And we just grabbed some genes from another fungus that does this, stuck it in, and it works. Just a little bit of computational optimization to make it work better in our yeast. Boom, solar powered, right? We wanted to explore how oxygen binding proteins affect the diffusion of oxygen through an organism, which can overcome anatomical constraints on size. Imagine that you get too big and all of a sudden there's a lot of oxygen out here. It doesn't diffuse well through dense metabolically active tissues. Well, animals evolved myoglobin proteins, which are the static oxygen binding molecules that make your muscles red. So we just took the genome sequence of a sperm whale myoglobin, which is very well studied and a very powerful myoglobin. We optimized it for yeast, ordered the genes, stuck it in yeast. They're meat colored and they have huge advantages.
And that worked. So, you can take a piece of it, and you just had a spare sperm whale lying around that you were able to.
So this is information. Somebody sequenced the myoglobin gene from sperm whale, and it was one of the first globins that was really worked upon because there's so much of it in sperm whale tissues. They store oxygen in it and then dive down like for an hour at a time. And so they're just using it as like a way of binding up oxygen when they're going to go on deep sea excursions. So
interesting.
It was like the biochemist's expedient way to get myoglobin. Just get some sperm whale and like isolate it from there, right? Like there's tons of it. So it's been really well studied. And so, we could just like go onto like one of these, providers that make synthetic DNA, again, change it a little bit so it works better in yeast, and then order it and jam it into our yeast, and it works. Like, so yeast are a really great model system.
It's insane. I'm not sure it's particularly common knowledge that there is this Amazon for synthetic DNA where you can just kind of browse and be like, maybe I want the stripes of a zebra. Or does that exist where there's just like lots of different random traits of things that are encoded by DNA? And
it's not a library. It's not a library like that. Like, you don't go shopping for what you want. You paste in the DNA sequence that you want created for you. And then they make that and they ship it to you. And then you basically can fairly easily insert it into the genome of your yeast. And that kind of service has been around for a long time. And it's used to be fairly expensive, but it's getting cheap enough that depends on the length of the protein that you're making, basically. But for something which is an average size yeast protein, it's under $1,000 to synthesize a gene. And often it ends up being cost effective compared to traditional cloning methods, because you're not spending a week of labor, doing these like complicated things where you're mixing and matching and cutting with enzymes and fusing things together and then putting that to a cassette and then flopping that into your yeast. Just like, okay, I could spend a week doing this, or I could just spend $500 and have it ordered and then just stick it in and it works.
So bizarre. Do you think that you could explain how that process actually works using simple language? Like how you actually get the gene of an animal like a sperm whale and you insert it into the DNA code of a yeast?
Yeah. So it's pretty straightforward. what you need is the information that encodes the protein, right? So the gene
Right.
is basically, it's the DNA bases in which there's a series of codon triplets. And so 3 DNA base pairs encode for 1 amino acid. And that is translated through ribosomes into a protein. And the protein is the sort of action thing which does stuff. In this case, it's a oxygen binding protein, which has a heme group in it and which grabs onto oxygen and can bind it or can facilitate diffusion through an organism. So we want to make our yeast express heme, right? And we can basically look up the DNA sequence of myoglobin. It could be a sperm whale, it could be human, it could be whatever you want. We chose sperm whale because
Natural
primarily
choice. Obvious
it's
choice,
a natural
really.
choice.
The
Yeah,
obvious
honestly,
one.
we also did some really nice like mathematical modeling of diffusion through organisms, and the biochemical parameters of how sort of the kinetics of like how oxygen is bound and how oxygen is released has been very well characterized for sperm whale myoglobin. So for us, that was the gene that we wanted. And then
Oh, interesting.
yeast have they have different biases in. Now you could just take the sperm whale gene and stick it in yeast and it might work, but yeast have this is a little bit in the weeds, but like the genetic code is redundant, meaning that there's 64 possible 3 base pair codes that can encode for an amino acid, but we only have 20 amino acids. So some of them have more than one codon that encodes for an amino acid. And different lineages have different biases in which codons they use for which amino acids. And that kind of matters because they have a pool of called the tRNA, the thing which will actually grab onto the amino acid and have the three base pair codon, which lines up with the DNA and has the amino acid on it. That's a pool floating in the cell. And if you use the wrong ones, then everything slows down because they're not abundant enough to actually power the synthesis of your protein. So we go through and yeast optimize any genes that we're bringing into our yeast by just kind of computationally like adjusting the codons to the ones that yeast like to use as opposed to the ones that other organisms might be using. And that's done by an algorithm. You could do it by hand, but there are algorithms that will do it for you. So you do that. And then you would also add like an antibiotic marker to the gene. So it's expressing the protein that you want, and it's also expressing an antibiotic resistance gene. You then take that whole gene and fairly straightforwardly, there's a method for inserting that into the genome of your yeast. And so you take that protein plus antibiotic marker dual action module, and you can you're throwing that at like 1 billion yeast cells, and you're just trying to get it into like a few. And so most yeast cells don't incorporate it and don't have it go into the genome. And how does it go into the genome? You have basically little flanking reference tags at the end of the whole construct, which will line up with the genome and basically will be flipped out for whatever's in there. It'll flip it out and flip your thing in. And so we'll replace like a standard gene that typically we replace in yeast that they don't need in our environment. So it has no cost to remove it. We know where it's going. We can check to make sure it went into the right place, and all that's easily streamlined in our lab. And you throw it at like 1 billion cells. You basically put the synthetic DNA in a test tube with a bunch of yeast that have been sort of treated to uptake DNA, and then have it sort of flip out and replace some of their DNA with the synthetic DNA. Then you put them all out on antibiotic-containing petri dishes, and you wait for any colonies to grow. And if a colony grows, well, it's expressing the antibiotic resistance gene. You then take that, you make sure that the gene went in the right place, you make sure that they're expressing it. And if it all works, that colony that grew from a single cell that was transformed now has like 1 billion cells, and they are all transgenic yeast. This is a method, like this basic approach has been around for decades in yeast. And there are fancier ways, like you've probably heard of CRISPR, which allows you to do more targeted types of changes. And if you're changing like individual base pairs, like you just want to make very precise edits rather than just kind of slamming something big in there, that would probably be the standard method there.
Very cool. Well explained.
Thanks.
The kind of a whistle-stop tour on synthetic biology and DNA manipulation. So how does that piece fit into your yeast experiments? Are you taking offshoots of these yeast and applying some synthetic DNA manipulation to them, and then kind of seeing what they evolve into, how that implicate? Like, Tell me about how synthetic biology really rolls into your methodology in the subsequent yeast generations.
Yeah. So I'll very briefly tell you what we were doing with our long-term evolution experiment, and then I'll answer that question because I never got there in the first time you asked me the question.
Please, yes.
So we started out with single-celled yeast, and we essentially, it's very trivial for them to form groups of cells. You just have to disrupt the function of one gene, and all of a sudden, a daughter cell can't fall off of a mother cell when it's mature and it's basically been budded off, and so mother cells remain attached to daughter cells, and they grow as these fractal groups. And for our long-term evolution experiment, we started 15 replicate populations where we actually knocked out that gene synthetically to give us a common starting point of a simple snowflake yeast. We call them snowflake yeast because they have this sort of fractal growth pattern. And then also we select for large size by letting them sink in our populations after a day of growth, and bigger groups sink faster than smaller groups, and it kind of looks like snow falling through our test tubes.
So that's an artificial selection pressure.
Exactly. So every day we impose artificial selection for fast growth rate. They have, basically they're in a batch of media, and if you eat the food faster than your competitors, you make more babies. You win, right? If you eat it slower, you lose. Then at the end of that day,
for yeast.
I guess I would probably not phrase it that way. But we're definitely not. No, I would say, okay, let me say it's definitely not eugenics yeast. We are not improving. We're not improving the yeast gene pool, and we don't have any ideas of what we're necessarily looking for. It's animal breeding for yeast or plant domestication for yeast, right? It's like agriculture. It's like we are selecting for traits that that's the classic directed evolution, selecting for traits that you want, and then letting evolution figure out ways to get there.
Gotcha. Yeah,
and in contrast to synthetic biology, evolution is a very good problem solver in navigating high dimensional space. You have a genome that has millions of positions. Mutations can come in and alter all of them, right? And, you have basically these explorations of many different paths towards a selective goal function. If you're doing synthetic biology, you have to kind of know what the right approach will be ahead of time, a priori. And sometimes that works, but often it doesn't, or maybe it works, but it's not the best. With directed evolution, like if you have a good payoff structure, which in this case would be fitness, right? Fitness is any trait that increases. Fitness is essentially the representation of your genes in the gene pool in the future. So any traits that increase the frequency of the genes that are in your body, those are adaptive. Those have a higher than one fitness, right? Any traits that are maladaptive would reduce the frequency of the genes that you contain in your genome in future generations. And there's only really two ways to change the frequency of future genes. You can either increase the survival of organisms that bear them, or you can increase their reproduction. But fundamentally, evolution is a birth-death process. There's a gene pool, you can add things to it, you can remove things from it. That's really it. All of life boils down to that very simple process.
Right.
And so, we basically, if you have a selective function that rewards faster growth or higher survival, then evolution can figure out really cool solutions to whatever your problem is. Okay, so we start out with these yeasts that are single-celled. We give them a mutation which makes them form these simple groups, and we put them through this daily selective pressure of grow fast. Then we randomly throw away about 90% of the population, and I'll tell you why we do that in a second. And then the remainders that are not randomly thrown away get this race to the bottom of a test tube, which selects for large size. So we're selecting for being big, we're selecting for growing fast, and that dilution step, that random throwing away 90%, that's to actually select for having a reproductive life cycle. One way they could break my experiment is just by growing really big, and then the whole yeast blob sinks to the bottom of the test tube super fast, and all biomass is transferred every day. And that would completely defeat the purpose of the experiment that we're trying to do, because we want our groups to grow, make babies, get big, and essentially have selection acting on the traits of these things in a multicellular context, have competition be between groups, driving group adaptation. And if you basically get rid of having a population of groups and just have one giant blob and all the genetic variations within one group, not between groups, then you can't really drive multicellular adaptation in this open-ended way. And by randomly throwing away 90% of the population, anything which grows and doesn't make babies is an evolutionary dead end. If you were as a group to grow every day for 3 days, you'd have a 10% chance of surviving the first day at best, a 1 in 100 chance of surviving 2 days at best, a 1 in 1,000 chance of surviving 3 days at best. Right. You can imagine like that's not a very good strategy, right? So you need to grow fast, make babies, and get big. And those 3 trade-offs together actually give you some really interesting dynamics when you start cranking that crank thousands of times.
Well, let's go into that then. What are some of the most important implications of your work? Like what's been one of the most interesting findings that has materialized over 9,000 generations, you said?
That's where we currently are. Yeah.
Yeah. And actually, one more point before you go to that. Am I right in saying that the mutations that the yeast are going through are still natural mutations? So that these are mutations in their own DNA. You're not manipulating those mutations itself. It's just artificial selection pressures, not artificial mutations. Okay.
That's right. The mutations are all natural. It's a funny thing. People tend to think that you would see lots of mutations in an experiment like this. And, we get about as many mutations in each population every day as there are bases in the genome, right? So these things there are tons of mutations entering every single day. And despite that, after, thousands of generations, you tend to see tens to maybe 100 mutations in a genome after 9,000 generations. The total.
Wow, okay.
That's very typical of these types of evolution experiments. Most mutations are costly, and you also have something where if there's 2 mutations that are both good, but one is slightly better than the other, it can kind of wipe the other one out. So you have to kind of the sequence and mutations arising in a genome that's already good is really important. And so it goes slower than you might expect, I guess. And to address that point you raised earlier about synthetic biology, we use synthetic biology for 2 major things. Our evolution experiment is driven entirely by natural mutations and this artificial selection pressure of grow fast, make babies, and get large. And, you need to do all 3, and they all trade off with one another. So it creates opportunities to get complex, break those trade-offs. But we can test hypotheses with synthetic biology. We can say, hey, do we think that these traits are important? And then we can re-engineer the ancestor using synthetic biology to do things that we saw happen in the experiment, to basically say, like, there's 50 mutations, but which ones are really important? Well, let me bring over these ones from the experiment to the ancestor. Oh, wow, they're doing exactly what we think they're doing, right? So you can test hypotheses. And then to bring in external tools, right? So we can sort of use our yeast platform to explore other molecular innovations and how they might affect the evolution of multicellularity, like understanding how the ability to use light for energy might overcome size constraints, or the ability to bring in oxygen through a tissue can overcome oxygen limitation, right? So like these are ideas that have been around, but they're difficult to test. And now we have a system where if you can engineer these things in, couple that with some more evolution, couple that with some really nice mathematical modeling, you now have like 3 different tools for testing hypotheses: engineering, evolution, and computation or theory.
It's very cool. You mentioned a really good general principle there that most mutations don't persist through generations because they're costly. They just don't confer enough of a fitness benefit to the organism. Presumably that's generalizable to all of evolution, in that every organism is evolving and lots of mutations are happening, but only a very, very tiny amount actually persist through, get selected for, and then become part of the gene pool. Would that be correct?
That's right. Yeah. Most mutations are either neutral, they have no effect, and rare neutral mutations mostly are just random walked back to 0. Right. There's this classic identity in evolutionary biology that the probability that a neutral mutation goes to fixation, or 100% frequency in the population, it's its frequency in the population. So if there's one mutation in one cell out of a billion and it has no effect on fitness, it only has a 1 in a billion chance of taking over in the population. All the other times, it will go back to 0. So most mutations are neutral or costly, very few are beneficial, and that's generally true in evolution.
Right. Are there any other general principles that you've discovered in your lab from yeast evolution that you can actually say this was true for even human evolution?
Ooh. I don't know how much our work really can overlap with humans, and that humans are, like, specifically human evolution is like a very special case. Like, you're not talking about animal evolution writ large, bilaterian evolution writ large, vertebrate evolution writ large. You're like going pretty far out on the tree to say like the difference between humans and other primates. And
Yes.
that's a very unique thing. Like, humans are very much a singularity in, sort of evolutionary history. We are unique in ways that are super cool, right? And what makes us so special is the ability to sort of, what makes biology special. Sorry, as a guest, you're noticing that I'm hard to corral, but I like to go off on, I love to sort of explain. I always go back
or do it, ramble, go
exactly.
on tangents, get out anything that you wanted to get out. Absolutely.
I start to go into the onion, and I'm like, wait, there's outer layers that are necessary to talk about. What makes biology special is that biology is chemistry that's been able to have open-ended Darwinian evolution. And that open-ended Darwinian evolution is what imbues chemistry with the information that underpins biology. And by open-ended Darwinian evolution, what I mean specifically is that you have things which have a life cycle, they reproduce. There's mechanisms of generating heritable novelty. So we have a genetic system which is binary information that's durable, that, you mutate a DNA base, and that can be passed on to offspring, right? And it can change the nature of the organism in a way that's heritable, and that affects fitness. Natural selection acts upon that and increases variants that are beneficial and decreases variants that are costly. And that process, there's a huge informational space that's open to that allows essentially open-ended evolutionary processes. We've had 3.5 billion years of open-ended Darwinian evolution, and we've barely scratched the surface on the potential possibility space. So chemistry isn't really that special. It's just that it's a polymer that has discrete states that has the capacity to allow for Darwinian evolution. But Darwinian evolution is not only something which happens in chemistry. It is effectively a mathematical identity. It is just something which, given the right conditions, will occur. If you have a system that does grow exponentially, have mechanisms that generate heritable novelty, which affect fitness, then it will evolve. And so you see this capacity playing out in information space, like computer programs, or in cultural evolution space, which is me getting back to the point that you asked about. So what makes humans special is that we've untapped open-ended cultural evolution, in that we have the capacity for ideas to spread from person to person and natural selection to play out on ideas, right? So if I have an idea for a better wheelbarrow, you can see that and you go, oh, wow, that's really cool. And that can spread from one person to the minds of 1 million people, and then someone can further improve upon that. And as a result, we're sitting here and we're taking advantage of the best ideas of, 50 billion people's lifetimes, or whatever it is, all right? But we are the inheritors of, cultural evolution running in an open-ended manner because we have brains, memory, and the capacity to encode information in an open-ended, arbitrary manner. We can describe things that are arbitrarily complex with our language because we have combinatoric language, which, just like a genetic code, allows you to essentially access unlimited information space potential. A combinatoric language does that too. And the communication capacity that other organisms that don't have combinatoric languages is much more limited. You have to have a different signal for every different intent, right? A dog can bark, a dog can, growl, a dog can look at you and, but each different signal has kind of one intent, as opposed to like taking 26 different letters and combining that to explain anything, right? Completely open-ended. So cultural evolution is what makes humans so freaking special. Right. And nothing that we do in our lab really maps onto that. Like that's the human magic.
But you were agreeing with me until I said the word humans, or you were agreeing with something until I said the word humans.
So good call. Yeah. Good memory. What we are looking at in our lab that is, I think, generalizable to the evolution of complex life or the evolution of multicellularity. Like, I want to be clear that we are not trying specifically to explain any one lineage of multicellularity. I say that because multicellularity has evolved not once or twice, but many times. Like, we don't even really have the right number because we keep discovering new multicellular organisms that we didn't know existed before. Every year we keep adding to the pile. But there's more than 50 independent transitions to multicellularity across the tree of life in all domains.
Wow.
And
That's cool. I
they
didn't know
don't
that.
do it the same way. Yeah. It's I think we all kind of are animal centric, but animals are also the weirdest and the least representative of everything else. Something like a plant is much more typical. Even within the green algae alone, multicellularity has evolved 25 times independently. But there is no one reason why organisms evolved to be multicellular, and there is no one sort of pathway that these organisms go down, right? In that they're evolving to form groups and then gaining novel multicellular functions to solve different ecological problems. And algae is doing it for different reasons than something which is swimming around trying to escape being eaten by a smallmouth predator. An algae might be trying to overgrow a competitor for light, whereas, something else might be trying to avoid being eaten by something which has a gape-limited mouth, and something else may be swarming over soil and trying to do so more efficiently, right? So there's different reasons why different lineages become multicellular, and they're mostly taking advantage of the genetic toolkit of their ancestor. They're mostly taking the genes that their ancestor had, putting them to novel multicellular purposes. This is like, if you look at the genetic toolkit of the ancestor of animals, they have most of the gene families that we use for development. They have them. We don't think they're using them. They're single-celled, more or less. We don't think that they're using them for multicellular development, but they've been repurposed for that, right? And so that sort of co-option story is very important. So every lineage has their own toolkit. Every lineage has their own ecology. Every lineage has their own reasons for becoming multicellular. That all being said, I think some things are universal. So those are the things that are not universal, right? What genes are involved? Well, it'll depend, right? But the things that are universal is this process of evolutionary dynamics. You have to have something where groups form and are capable of open-ended Darwinian evolution. Groups have to have a life cycle. They have to be able to reproduce in an exponential manner, right? So, you put one in a test tube, you come back a day later, there's 1 million, right? And now all of a sudden they're out of resources and now they're in competition, right? If something isn't reproducing, it's non-Darwinian. You have to have a mechanism for mutations to generate new multicellular traits. And in fact, these traits have to be dependent upon those of their cells, but different. Philosophers would say non-commensurable. And what do I mean? Any trait of a multicellular organism, you can even imagine yourself in this scenario, has cell-level causation, right? Your maximum muscle twitch speed is a function of the way that neurons communicate, with electric impulses across cellular bounds. And you could imagine that there's some mutation which changes the kinetics of a membrane binding protein, which allows it to transmit information faster. That means you can run faster, right? There's a cell-level mechanism that underpins the multicellular trait. These two things are coupled. The mutation's only directly affecting the cell level, but it's giving rise to an emergent multicellular property, right? And
Right.
and importantly, they're not the same, right? So like the cell level trait is like some, I don't know, like ion sensitivity of a membrane of a voltage gated channel, and the group trait is, did you survive the crocodile attack? Can you twitch away fast enough when a predator tries to bite you, right? And this is true for essentially every multicellular trait. Information arises at the lower level, but selection is acting on a higher level emergent multicellular trait. And then can that essentially underpin open-ended Darwinian evolution of groups, allowing them to, over millions of generations, embed more and more and more information in that multicellular space? That process is general. We believe we have no idea how it could exist outside of that paradigm. And yet it's really hard to study that paradigm. It's really hard to see those dynamics in nature because essentially the evidence of it in extant lineages like animals, it's gone. That happened 800 million years ago in animals. When we're talking about origins, like simple groups becoming more complex, right? We just can't really see those beginning steps. And then other things that we think are pretty universal have to do with trade-offs. So as organisms get bigger, bringing in resources becomes harder and harder. Like there is no multicellular organism that's large that doesn't have some form of a circulatory system. Because otherwise you would, diffusion would just take too long and inner things would be completely metabolically inert. So, plants, animals, sea sponges, like these things all have some mechanism of moving the outside environment through them, allowing them to overcome diffusion limitations. So in our experiment, our yeast have surprised us repeatedly at figuring out how to solve some of these fundamental problems in ways that are much simpler than how it's done in extant organisms. So,
Interesting.
like, for example, so over the first 3,000 generations of evolution, our yeast go from being about 100 cells per group, very, very brittle materials. They have these fractal branches of cells where if you break any one cell branch, it's like cutting a branch off a tree. Like the downstream branch just falls off. It's not connected to the rest of the group except through mother-daughter connections. And so it's a very bad material. The material is only one cell-cell bond from ever fraction. You only have to break one bond and you break the group. And we're selecting on them for large size, and they have to figure out how to become tough, which is it's not a challenge that they've really had to deal with in their evolutionary history. And so, at least not in the way that we're giving it to them. And so, at first they evolve, these like more elongate cells, which reduces the amount of strain in the interior from cell-cell overlap and packing. They evolve bigger and stronger bud necks, which, increase the amount of strength it takes to break these two things apart. And those give you moderate changes. We're talking about going from 100 cells to maybe 1,000. But then all of a sudden there's this jump where they go from 1,000 cells to 500,000 cells. And the organisms go from being weaker than gelatin to as strong and tough as wood.
Wow.
I got to get some water.
That's an incredible leap.
And the way that they've done this is that they basically held off the groups breaking apart long enough that the cells could grow around one another. And once the cells wrapped around one another, rather than being like a tree, they became like a vining entangled system. Once they're wrapped around one another, if you've ever tried to like rip a vine off your shed or something, it's extremely difficult because the forces on these chains of cells are propagated throughout the entire tissue. And now to break one branch, you basically have to break all of them. And they did this without any mechanisms of multicellular development, the way that a regular multicellular organism would have. Extant multicellular organisms have a lot of developmental regulation. They have developmental genes that coordinate pattern expression and morphogenesis at the multicellular level, which mutations can arise in and change their behavior, and that gives you new multicellular traits, which gives you this really nice connection between multicellular traits and cell-level information. Our guys don't have that layer of regulation. They're just, in the beginning of the experiment, they don't even know they're in groups, right? It's like cells are growing as single cells. All of a sudden, they're in groups. And now we're selecting on those traits of groups, and they're able to basically figure out mutations come in, which just, randomly change the behaviors of cells. And when those are good for groups, they make them bigger, they make them tougher. They're really adaptive, and they sweep through the population, they fix. And you can see them basically figuring out these solutions to things like toughness without the developmental layer that we kind of thought was necessary. And they're doing it by kind of harnessing the emergent biophysics of cells living in groups, which is cool to watch them sort of just bootstrap their way up, right? They're very simple, and yet they're figuring it out, and they're doing it quickly. And
are these yeast, when do they become multicellular? Like, is that fractal
Great
configuration
question.
snowflake, is that multicellular, or is that just a really big pile of the same cell?
I wish we had a better series of words in the field. This is the single largest source of confusion in my field is, what does the word multicellularity mean?
Right.
So multicellularity is a noun, and multicellular is an adjective. And really, the one word multicellularity is like, it's a noun, but it's really the adjective modifying three separate nouns. And these three separate nouns are three different things. It could be a multicellular group. Some people, when they say multicellularity, they just mean there's more than one cell in a group. It could be a multicellular Darwinian entity. And this would be, a lineage which may be simple, but is participating in this process of open-ended Darwinian evolution. You have groups that reproduce, that have heritable variation, selection acts upon it, and drives adaptation at the group level. Or it could be a multicellular organism. Most people kind of mean the latter, but not everyone, right? And an organism is not a description of evolutionary process, it's a description of function. An organism is something that has functional integration of parts, high cooperation within the group, low conflict, and organisms evolve from Darwinian entities, right? So like the way that you get organismal traits is by making these simple groups Darwinian and allowing them to essentially gain adaptations. And so when someone asks, when does it become multicellular? It's really actually like you need to go to each of those three and say, like, initially, it's a multicellular group. Bringing in one mutation that prevents daughter cell separation makes it a multicellular group. It turns out that it also makes it a multicellular Darwinian individual because those groups have a life cycle. They grow till they run out of space. They break a branch. That branch pops off. It's its own baby snowflake. Those have genetic bottlenecks in them. Like every time you break a branch, that bottom cell is the clonal ancestor of everything downstream. So if this has a mutation, then its daughters, its granddaughters, its great-granddaughters, everything in the branch has that mutation. So mutations segregate between groups. Natural selection is acting on the traits of groups, driving group adaptation. That happens right away. So they're also Darwinian individuals or evolutionary units right away. And then when do they become organisms? And so, this actually really brings it back to the philosophy of individuality. What do we mean by an organism, and at what point do we have enough evidence that something is an organism?
Right.
And there is no like rule book here. But typically, what I want to look for is the evolution of traits at the group level that don't really have that are novel and meaningful and adaptive, and also heterogeneity, functional integration. So you want the cells to be dividing labor, or at least specializing. And you want cells to become interdependent, such that they can no longer leave the group and be happy on their own, which is certainly true of most multicellular organisms. I have skin cells, which protect my body from the outside. They're not reproductively on the line of descent. If I scrape them off, they're dead. They're not like amoebae, which eventually form a tiny will, which, I don't know, I could get to help write grant reports or something, right? Like they're just gone, right? Like, and so those are actually the things that we're looking at. Those are happening in our system. Those are unfolding as the system continues to evolve. We're beginning to see new multicellular behaviors that break trade-offs and are really exciting. We're beginning to see what we think of as cell specialization and distinct cell types from one genome. And we are seeing the lack of ability to bring these guys back to single cells. Initially, we can make them go back to being single-celled, but after sufficient evolutionary time, we really cannot make them grow as single cells again. So we're kind of seeing this entrenchment in a multicellular state, cell differentiation, and even cell sacrifice, where a fairly large proportion of our cells are giving up their life, which is clearly costly to the unit of the cell, but we think it's beneficial to the unit of the group. And this type of sort of cell-level altruism in the service of the success of the group is like a classic sign that cell-level fitness isn't really the focal point of selection. It's group-level fitness. And then, any ways that cells can facilitate, better groups, that pays off.
That's so interesting. I'm remembering back to my evolution lectures, and at least at the time, there seemed to be some debate on group selection. And I think this was mostly related to altruism, and if an individual embodies altruism and it benefits the group, will that trait be selected for basically on the benefit of an entire group? And there were some advocates of this idea, and there were some people that were against it. Is this the same group selection idea that you're talking about, or is it different?
We're talking about a slightly different one in that, and by the way, I sort of wish the field had gone a different way with that debate. I think there was a lot of argument about something that is fundamentally, we should have a pluralistic stance rather than a group selection is right, inclusive fitness is wrong, or vice versa stance.
Right.
These are different ways of conceptualizing the way that the structure of selection acting on traits and genes. And they both have cases where they are the best explanatory mechanism for the system that's under consideration.
Nice.
Now, one key distinction between what we're talking about, which would be sort of the multi-level selection of groups of cells, and like a classic evolution of altruism in a group context argument, is actually that in the classic argument, those are framed around selection acting on the traits of individual particles within the group as a function of their effect on the group as a whole. So you would have altruism of a particle within a group based on group productivity or group survival.
Right.
But this is always seen as like coming at the cost of that cell within a group, and whether or not it paid off really depends on the group structure. Is it mostly with other altruists, or is it mostly mixed with other non-altruists? The strength of selection, the trade-offs, and when all that math is done, it's either beneficial or not. And that's just a mathematical identity. That's not something that you can really argue with. It is just true or not, right? It just is the world. But in the case that we're talking about, we're actually talking about a different sort of flavor of multi-level selection. And I think one that even very strong partisans against group selection would kind of be happy with, I would hope, which is that any group of cells, any organism, which in that classic evolutionary biology parlance would be an individual, like a redwood tree or a dog or a mycelium grown from a single spore, right? Those are multicellular organisms. They're groups, and they are, in a sense, individuals in the way that natural selection is acting upon them. They're rounding that group of 1 million cells up to just one individual, but it's really selection acting on the traits of the group. And why this is so important is that we're not talking about natural selection acting on the traits, like we're not talking about cells as the units of selection within a group, as a function of how they affect the fitness of the group. We are talking about natural selection acting on the traits of the group itself, which of course have cell level causality, right? Like I gave you that example earlier about like running speed and nerve firing rate or something, right? All traits of multicellular organisms must have cell level causality. That's just the way, because they're made of cells. And these traits, they are often, while they're connected, they're often distinct. So pretty much all of the group traits that we were talking about in the old evolution of altruism world were just the average of the traits of the cells within the group, the average of the traits of the particles in the group. The group trait was like mean productivity of cells. Here we're talking about group traits that are different from those of the cells. Here we're talking about what's cell shape, what is group size, what is cell electric impulse firing, what is group muscle twitch speed, right? And so while they're causally linked, they are non-commensurable. The group trait isn't just some simple additive function of those of the cells. It's some crazy nonlinear function. And so why does this make the most sense in a multi-level selection framework? It would be very cumbersome to use an inclusive fitness model of like really anchored around the cells in an organism and their behaviors and how that affects natural selection acting on their traits. If you tried to make a model of an organism kind of from the cells up, you could do it. There's no reason you couldn't do it in the same way that I could use quantum physics to describe the behavior of my water bottle, but classic Newtonian approaches are just fine, right, and don't have to get into all that lower level detail. But you can of course look at it from the bottom up as opposed to sort of the top down. But a multi-level selection model here is saying, all right, I have these groups of cells, they have these traits, selection is acting on those traits, it has some heritability, and I have some resulting evolutionary output. While classic evolutionary biology say that's an individual level model, it's really a group selection model. It truly is a group selection model. So I would hope that even people that don't like group selection would realize that and be like, yeah, actually, that does make sense.
I think it does. I think that's quite clear. What do you think are some other big evolution myths that are very common that you hear that just make you grind your teeth because you're like, it's just not how it works? What, even down to the ones that are very silly?
Well, there's this idea that evolution always increases complexity, and that's a very commonly held one that, this goes back to pre-Darwinian ideas, like Lamarck, this great chain of being.
Right.
Life starts out simple and it gets complex. And there's I understand that because if you look at the history of life on Earth, it did start out simple, and some lineages have evolved to become really complex. And so there's sort of this interesting, it kind of maps to like the intuitive experience here, but that's not because there's any prerogative for life to become more complex. That's because it's building upon prior successes, and there's ecologies that are available to organisms that are expanding the frontier of complexity. But most lineages don't evolve to become complex. Most lineages stay single cell, right? And it's, it has to make sense to explore a new multicellular ecology. It has to be stable. If after a million years, single-celled organisms have higher fitness in that niche, then you're not going to have persistent multicellularity. So that's one, right? And you often see losses of complexity, right? So in some lineages, you see losses of multicellularity. In some animals, you see losses of eyes if they're no longer using them because they're in a cave or something. So there's no reason life has to get more complex, and we shouldn't expect that it necessarily does.
That's a good one.
If you start simple, there's always opportunities to get more complex, right? So that does happen.
I think people have this sort of Lego building block of idea of evolution, where like evolution is just building Lego blocks, consecutively building on top, and it's always just becoming this more expansive structure. What's an example of what you just said, where it goes from multicellular to unicellular? I've never heard of that before. How does something go from that much more complex to less complex?
There's very, very little work that's been done on this topic, which is interesting.
Right.
So some lineages have left no single-celled, free-living descendants. So animals, plants, think the brown algae. I don't think there's any free-living, single-celled descendants of those lineages. So they have not reverted, which is kind of spectacular because just think about the amount of animal biomass or plant biomass that's around, right? Like, we're talking gigatons of cells, and basically, and we can revert them we can make them grow in the lab as single cells. You can get immortalized cancers that grow just fine without that multicellular context. But this does not, to our knowledge, happen in nature. And there are some interesting parasites that are derived from animals that are now single-celled parasites, but they still need the sort of enclosure of an animal body to be successful. They're not free-living single-cell organisms. Interestingly, fungi have made many reversions back to unicellularity. Some green algae have done that. And I don't think there's any papers like talking about why we think this is, the conditions under which it happens. We're just about to submit one on entrenchment. We have a bioarchive paper. You can read the last version of it, but like this week we're submitting one on how division of labor entrenches a lineage in multicellularity, that once you have organisms in which the cells are actually dividing labor amongst themselves, it becomes difficult to go back to being single-celled because now they're basically really unfit for that single-celled environment, right?
Interesting. They rely on all of the systems and all of the different components of the system, so it's very difficult to go back to not relying on them.
Exactly.
Right.
But it's, in our system, it's both mutationally harder. There's fewer opportunities, and then ecologically harder. That they are worse at doing what their ancestors. They're basically being outcompeted by all the things that didn't become multicellular. So their fitness as a single cell is much lower.
Is this really how, if you thread that needle all the way out from basic multicellular all the way to humans, can you kind of summarize that as many subsequent steps of spreading out the reliance on the system onto more and more components, and that's why you get this progressive step? Now, you say it obviously can jump backwards, but is that one of the most fundamental sort of points in the evolution of complex systems, is that it's becoming reliant on more subcomponents, and then more, you need a liver, and suddenly you need a heart, and suddenly you need a stomach, and then once you become reliant on those things, you can't go backwards, or it's extremely difficult to go backwards?
I think this is an area that people need to research more, right? But I think that's a pretty good summary of kind of where people are at. That division of labor, and I actually don't like the term division of labor because that sort of presumes that multicellular organisms are doing the same thing their single cell ancestor did, just more efficiently. And there is some of that, but even more fundamentally, you have the opportunity to evolve into new ecologies and have new morphologies that are fundamentally different from what the single celled ancestor had. And I think a lot of like cell specialization is less you can think of it as dividing tasks. If you think of the organism as having a task, right? My skin, my heart, my eyes, they all have tasks and they're dividing labor.
Right.
But you can also think of it as like the evolution of just new morphologies, right? That give rise to new organismal properties. And there is a long history of trying to sort of look at the complexity of multicellular organisms, like describe that by the number of cell types they have. It's funny because just like any other binarization problem in biology, what a cell type is, once you start doing that, you actually have to answer what a cell type is and how, and that's not trivial, right? So like, it's really interesting how often this problem comes up in biology. I don't know if there's like a sort of physics or math equivalent. I imagine that like you have some researchers being like, okay, what exactly is the wavelength where like blue becomes green? Like just like, I know that there's blue and green, but when does it transfer? Like, I need to know. Like,
Yeah.
having some like coffee mug throwing argument about like exactly what the wavelength is. We have the same problem in biology.
Right.
As soon as someone brings up cell types, it's just like, okay, great. Now we're going to talk about cell types for an hour.
You can't agree on it.
And I'm not saying it's wrong, but like describing things categorically when they're fundamentally continuous is really hard. And I don't know that there's like an objective way to do it.
Right.
But that being said, your intuition, I think, is right, that like organisms can become more complex by having a greater number of possible subcomponents that do specific things. And certainly animals have way more cell types by probably any measure than any other lineage of multicellular organisms. Yeah. And once you're kind of locked into that ecology, I think it can become very difficult to go back. And maybe one of the reasons fungi have had such an easy time going back is that when hyphal networks, like the real complexity of fungi is in building a fruiting body, so a mushroom. That actually takes a lot of spatial coordination and morphogenesis and regulation, and those are complex by any definition. But when they're just growing as like a hyphal network in the soil or in the water, they're just chains of cells, and every cell is in contact with the environment, and they're basically eating just by dissolving food in the outside and osmotically absorbing it through their tissues, right? And so that ecology might also be one that lends itself to reversion, that like a single-celled yeast can make its living in basically the same way. It's just no longer touching other cells, right? Like there are real advantages to forming a hyphal network. And the main thing is nutritional arbitrage. If over here is too much carbon, but not enough nitrogen, and over here is too much nitrogen, but not enough carbon, cool. You've connected the dots. You have a railroad system. You're flowing stuff back and forth, and the whole thing can grow, right? But you don't need that. That's an advantage, but it's not a need to have. And if you're doing what normal yeast do, which is hanging out until there's a jackpot of food, like someone dropped a cherry tomato on the ground, and then all of a sudden you just explode in that environment, right? I don't really think you need a hyphal network. It's probably not very advantageous. And so maybe it's easier for something that has that ecology to go back.
Right.
Whereas if you're an animal, and you've built up all of this insane developmental complexity, you really walled yourself off from the outside world, it may be quite a bit harder.
I definitely want to go into those networks later because there's just so many cool things about, stuff like mycelium networks and connections with intelligence and all of that. I want to touch on that later.
Nice. Cool.
But I want to go, we haven't mentioned LUCA yet. So I want to dive into LUCA. What is LUCA?
Your accent caught me off guard. I was like, I don't know what LUCA is either. You mean the last universal common ancestor?
That
LUCA?
is what I mean, yes.
Excellent. So LUCA is an acronym you hear when people are talking about the origin of life, in that it stands for the last universal common ancestor. And this is this idea that basically there's a phylogenetic bottleneck, meaning if you look at the sort of tree of life, right, you can coalesce all extant life back to a singular bottleneck point. That doesn't mean it was one organism. It doesn't even necessarily mean it was one population, because the way that we think about biology now is actually probably quite different from how porous the sort of genetic systems of early organisms were. But it was sort of like there is a coalescence point in which all of the sort of molecular biology of life can be traced back to and all diverge from that common point. And they're likely were.
Fact. I think it's worth restating just how fascinatingly cool that is. That is bizarre.
It is
that
so
you can
cool.
track everything back to basically one organism.
So one, let's call it population.
Okay.
because it doesn't necessarily have to have not all of the machinery that was in LUCA was in a singular organism. It could have been a population of a bunch of different related organisms that are sharing genetic information via horizontal gene transfer, and that population then evolved into life as we know it, rather than a singular genome containing all of that.
Right. Okay, so that's a misnomer you've already corrected for me, because I had a singular organism as LUCA, but LUCA is more like a population of gene transferring organisms that are working together in a super ecosystem like the human body is, with the amount of bacteria and things in our...
I don't even know that I would imply that it was necessarily highly cooperative and organismal. More that if you imagine that you have an ecosystem of bacteria-like organisms in which they all share a common ribosome. They all share a common genetic code. There are certain things that we have, right, that are legacies of that last universal common ancestor. And the core molecular biology of cells is relatively little changed from 3.5 billion years ago. We understand the evolution of the ribosome through Lauren Williams's work really well. You can see how you kind of add on protein and RNA to sort of this core catalytic, substrate. But that core really hasn't changed structurally in almost 4 billion years. It's outstanding, right? We have a common genetic code. And if you think about the genetic code, what do I mean there? It's the translation from those, this goes back to the beginning of the talk, those amino acid triplets like AAG into, those base coding DNA residues, a triplet of 3 DNA bases, which is translated into a amino acid, which is the building block of the proteins, right? That actually goes through the ribosome. And to a certain extent, the genetic code is arbitrary in the same way that our dictionary, which takes letters and turns them into words, is somewhat arbitrary, right? We have lots of different languages, and there are ways of encoding information, but they don't have to be a certain way. To a certain extent, the genetic code, the canonical genetic code, is not arbitrary. It's actually very optimized for minimizing a number of different types of errors that occur during translation. It is much better than a random genetic code. Computer scientists have been all over this. So the genetic code has elements that make it look like it's evolved, but it's also like the least evolved thing in the entire history of biology.
Right.
All organisms have the same genetic code, barring a few tiny changes in different branches, mostly within obligate symbionts that are probably kind of buffered from the catastrophic consequences of changing your dictionary. Once you have a strong dictionary, you change anything in there and nothing makes sense anymore, right? So it becomes very, very locked in. So there's a number of these things that are like deep homologies. Everything more or less shares the canonical genetic code, barring a few tiny tweaks. Everything shares ribosomes. Everything shares, the basic sort of fundamental structure of the way that cells work. Core metabolism is effectively the same. And we can trace those origins then back to a common ancestor. When we say the word common ancestor, we think of individual, the way that our ancestors are individuals, but it's better to think of it almost like a population of interacting things which contained all this stuff, which then continued to evolve and diversified into all the myriad life forms that we see today. But it wasn't just one organism. And this could be a population of things that are competitors, are cooperators, are pathogens. Like, it doesn't have to be just like an organismal-like population. Just think of it more like an actual real world ecosystem.
Right.
With all the different ecological roles that would entail.
Where does the best evidence say that, when was LUCA alive? How long ago?
So the best evidence is like the canonical answer to that is 3.5 billion years, but it's also like that we should be a little bit mindful that it's really hard to make good estimates going that far back.
How do you even what even is the evidence for that? I don't even understand how you can have evidence that it's 3.5 billion years old. And I read I'm curious if you saw a paper that came out this year that dated LUCA to 4.2 billion years, which is basically like 100 million years after the Earth was formed. I can send it on to you afterwards because I was making.
I think I actually did read that.
Yeah,
I'm on a committee for a student who's working on pushing back these deep time datings pre-LUCA. So she's actually trying to work with bacterial and archaeal genes and push back their coalescence to she's essentially arguing that bacteria and archaea have much deeper roots than we think, which is pretty exciting. It's hard. It's really hard. You can use a variety of molecular clock tools to try to estimate that, and it's really hard because a lot of the classic assumptions of molecular clocks, like fairly consistent rates of molecular change, probably don't hold very well, especially when you start just going back so far, right? It does, on the other hand, kind of line up with fossil evidence. So we actually have like legit fossils of bacteria-like organisms that date back to 3.5 billion years from hydrothermal vents. Like they look like cells. They have they are depleted in 13C. Sorry, yeah, 13C. So carbon is normally 12. It has a molecular weight of 12, right? But you have like this stable carbon isotope that's got an extra neutron in it, behaves just like regular carbon, but biological processes like tend to discriminate against it because it's just like kinetically too big to like get grabbed by enzymes. So it tends to get excluded. And so biological processes fractionate against 13C. So we can actually look isotopically, like in these 3.5 billion year old microbial looking fossils, they're also 13C depleted. So it like really looks like, looks like cells.
How does a bacteria fossilize? Because doesn't bone and teeth, and it's usually hard material backbone, sort of skeletal backbone that usually fossilizes. How do you fossilize a...
Definitely. Yeah, so I mean, anything can fossilize. The harder things are, the easier it is, right? There are lots of there's lots of microbial fossils. You just need the right conditions for it.
Cool.
And these were found in like
glaciers and things like this, is it?
Hydrothermal vents, deep sea hydrothermal vents.
Cool.
Yeah, but there are actually you should take a look at some of my favorite fossils are these like 1.6 to 1 billion year old eukaryotic fossils that have been found in like crazy places like, the outback of Australia. And these like 1.6 billion years ago, you have these intercrotonic basins where you have these like shallow seas, and they have such fantastic fossil preservation that you can like dissolve away the rock with hydrofluoric acid, I think, if I'm remembering some talks I've seen. And you get these fossils where you can actually see like the cells, and it looks like you're looking at them under a regular microscope. You can see that some of them are multicellular and like I have cool appendages, and some of this preservation is, or Google the Daoshantu fossils from China. They're like low 600 million year old fossils where, they look like animal embryos, but nobody has any idea what they really are. But you can see like subcellular features. Like the level of preservation is just outstanding.
That is very cool.
It's so cool.
What can we learn looking back that far? Again, I'm curious, like how do we connect 4 billion years of history to what's happening now to make advancements in science, to make advancements in any field? What really are the learnings from LUCA, how LUCA lived, who his friends were, what his environment was, all of those different things?
That's a fantastic question. And I'll preface this by saying I'm not the right person to ask because I don't really work on LUCA. I don't work on super deep time stuff. Multicellularity is pretty old, but like it's its own thing, right? And I think we can all understand why. Understanding how...
LUCA was multicellular though, was he? Or was...
No, single cell.
Okay.
If it was multicellular, I wouldn't be super surprised given how easy it is to evolve multicellularity, but we don't really have any reason to believe that's true.
Right, okay.
Yeah. So there's a fundamental sort of like knowledge component to this, right? Like if we want to, I think the origin of life is, if not one of the biggest scientific unknowns, right? And we've made a lot of progress on it in the last several decades, but it still remains something where we're making plausibility arguments rather than arguments about how it exactly happened, because there's very little information that remains to today that would actually allow us to piece together how chemistry became Darwinian and evolved into the life that we know today.
Right.
And this is, it's a fundamental, just pure knowledge-based scientific question, right? Like, how limiting is the origin of life, right? What are the conditions under which it arises? If you understand how it happened on Earth, you get information about the geological components that sort of played into this. Like how did it happen? Are the conditions under which it occurred special, or are they super common in planets, right? People have made arguments in both directions, but I'm actually I'm very sympathetic to the it's not special or particularly hard camp. That you have planetary scales of time and space. You have fundamental thermodynamic conditions in which energy is being dissipated from planetary cores out into space, essentially via the oxidation of metallic cores, allowing essentially protons to flux from the core out into the proto-oceans and eventually be lost out into space. And so you just have essentially battery-like conditions where life has a niche for dissipating thermodynamic disequilibria faster than non-life could, and that is coupled to the spread of those chemicals, and that sort of can power the origin of life. But how exactly does that occur? It's something that we still really are working on.
Right.
And so, in terms of big questions, there are fewer that are bigger, and it has a lot of I think extrapolation to how common is life in the universe. I tend to think that it, at least relatively, organisms up to humans or whatever, excluding humans, it may not be that hard. I really don't think that any of the priors that we know about are particularly large bottlenecks. You talk about the origin of life itself, it happened early. If it's 3.5 billion years, that's probably a conservative estimate. Origin of life probably was closer to 4.2 billion years. And that's not very long after Earth became habitable, cooling down. It's only 4.5 billion years old. In the beginning, it was a big ball of molten lava, right? And, to the extent that what are the other sort of big hurdles, right? So it's getting cells which have DNA, something which allows you to have DNA-like encoding of information, ribosome-like use of that digital information for function. Having a cell membrane is critical. That allows you to compress a whole bunch of stuff into one space. That makes cells very effective evolutionary units because they have the information and the capacity to do a lot of stuff. Cells are they're groups in a group selection model. They're groups of subcellular replicators that are now all selections acting on their traits as a whole, right? Eukaryotic cells are also groups. As much as we don't think of a single eukaryotic cell as being multicellular, a eukaryotic cell is a complex cell, right? Which has a nucleus, which has a bunch of organelles that bacteria and archaea don't have. And it resulted from the symbiosis of a bacteria and archaea, right? The mitochondria that are in your body are formerly free-living bacteria. They still have their own genome, although it's highly reduced. They still have their own...
Sorry, it's just another fact that it kind of... Because you live this world so often that you say a lot of things that I think are worth restating because I think they're just staggering. So every human on Earth has a bacterial line of genetics, which are our mitochondria.
Yep.
I believe we always inherit them from our mom. That's correct?
Correct.
You always get them from your mom. That's just one of those things that when I learned it, I was like, everyone should know that. Everyone should know that, but not a lot of people know that. That's profoundly interesting. So that was the great leap is what they call it, around 2.3 billion years ago, this phagocytosis event where one big cell ate a smaller one, and that smaller thing is now the powerhouse of all human beings, and not just human beings, but all animals and plants, right? And basically everything.
Everything. Not everything. All eukaryotic life.
All eukaryotic.
So
staggeringly interesting.
There are cool fringe things where some eukaryotes have lost their mitochondria, but most eukaryotes have preserved this symbiosis for 2 billion years. And this is one of these things when people are freaking out about AI alignment and how do we align common interests of things that are fundamentally discrete? This is a solved problem in biology. We have a 2 billion year old cooperation that is super stable.
Good point.
And it could probably last till the death of our solar system.
It's an interesting
got billions of years to run, right?
And how similar are they really? How similar is that mitochondria to what exists now? I know I'm asking all these questions about what happened 2 billion years ago, and you're going to preface all of them with we don't really know. But how conserved is that mitochondria from 2 billion years ago to today?
Oh, it's super different from its ancestor, right?
Super different.
Its ancestor would probably have had a genome size on the order of 3 million base pairs, which is typical bacteria. And its current genome size, depends on the lineage, is around 25,000 base pairs. So
that's today.
Okay,
25,000.
Most of
We've
its
stripped away the parts that aren't needed.
Oh, even better than that. Most of the genes that used to be in the genome of the mitochondria are now residing in the nuclear genome. So
interesting.
We've taken control of the tools of gene expression for most of the things, and they are under strict host control.
And it's still utilized within the cell. We haven't stripped it away and lost it because that symbiotic ecosystem is more fit or confers more fitness than ever losing those compartments.
That's right. Right.
That's so cool. So interesting.
Yeah.
I didn't know that actually. It's really cool.
It's the same for chloroplasts. So all plants are photosynthesizing with cyanobacteria that became chloroplasts.
Do you think I have a question about the intertwining sort of yeast cells that you were talking about earlier that are becoming these sort of vine branches? Do you think that's almost like another phase shift, another leap to what back in the evolutionary timeline could have been a more complicated organism? Because the first thing I thought of, what came to mind when you told me about this, gelatinous basically substance, and then when it intertwines together, it becomes, I can't remember what exactly you said, but a lot more rigid. Was
strong and tough as wood.
Yeah, exactly. Was skin. That it's sort of becoming a little bit like skin. And I'm probably jumping there, but do you think that this could be what you're creating in the lab is actually a phase shift in evolution, and you could get something that's just a bit crazy?
It's possible. I don't know that skin is as we think of skin is something which I think is super likely in this particular system, but other forms of functional integration seem to be happening. So I'll tell you what one of my graduate students is currently working on. It's not published, but it's something that, it's his main PhD project, is looking at how cells share resources between cells in our group. So normal yeast have they'll store lipids, fats in little droplets, and when they divide, daughter cells get a little allotment of the mother's kind of random allotment of her cytoplasm, which contains lipids. By 5,000 generations, our yeast have basically pushed all of those. They're very polar now. Rather than being kind of randomly distributed in the cell, these lipid droplets are actually associated with some of the subcellular catalytic machinery of our cell, and they are shoved just to one very end of the cell. So the cell looks like a hot dog. They're stuffed at the very tip of that hot dog.
Okay.
And then when that cell divides, the first daughter gets most of those resources and they flow into her, and second and third daughters get very few. And you can actually have sort of this transgenerational flowing of resources from mother to first daughter, mother to first daughter, over and over and over. And what we think that's actually doing is sort of two things. And these are sort of the hypotheses we're testing now. So first of all, the more resources your mom gives you, the faster you grow. So they're expanding the rate at which they grow in a straight line before branching out sideways. And pre-entanglement, so the early snowflake yeast, that makes them get bigger because it reduces cellular crowding in the interior, which causes them to break apart. Post-entanglement, they're really tough. That's not really a worry anymore. What we think is happening is that these groups often will kind of break and get cracks that don't fully separate. And if they're going to grow and not just fall apart, which again, we're selecting on large, tough size, then they need to have a way of basically healing that wound and growing branches across that crack and then re-entangling and re-stitching the material back together.
Wow.
And if you can basically make that first line of cells grow faster than all the side branches, then you can actually, we have really nice computational models that show this. You can actually stitch across that gap, side branch, and knit the thing back together and re-entangle, and you can heal wounds. And so you can sort of imagine you have this scenario here where there's a selective advantage to being big, a selective advantage to figuring out how to make a tough body. They figured out entanglement. It's not perfect. It's good, but it's not perfect. You still have these deep cracks that occur. And now there's an opportunity to begin evolving functional integration where cells are now changing how they allocate resources, which can potentially solve this problem. I'm saying a lot of potentially because, I'm saying I'm hedging a lot because, again, It's not a published paper. It's work in progress, and we may be wrong. But it's very exciting to sort of begin seeing how they solve these fundamental problems of multicellularity. Here's another cool one.
Please.
I was at Woods Hole teaching a couple years ago, and we brought some big snowflake yeast with us. And we'd actually been working with some awesome collaborators from India, and they were like, "We think the yeast are actually generating really rapid hydrodynamic flows, like the way that sea sponges like shoot media, like suck in water and shoot it out their top. Like we think snowflake yeast might be doing this. Like they had gotten strains from us, and they were looking at like scaling laws and how like group body size affects growth rates, and we expected to see something that has to do between the ratio of surface area to volume, and the bigger they get, the slower they grow, because internal cells are nutrient limited from diffusion, and they're just sitting there waiting for food to arrive. And we looked at, a 20,000-fold range in group size, and the scaling coefficient was like 0.97. Like it should have been like 0.7 or points, should have been really quite low. And it was like, basically these things were growing at the same per capita growth rate when they were 500,000 cells as they were when they were 100 cells. So we threw these things on some really fancy microscopes, and we began to watch them actually acting like sea sponges, like sucking water from the outside and shooting it out the top, like really fast. You could see it with the naked eye. And turns out it was spontaneously driven flows based on the buoyancy of media inside the cluster compared to media in the environment outside it. So the cells on the inside ferment, they use up their food. In fact, they make some alcohol out of it. And that waste product is much less dense than the water outside. And that means that just like if you turn on a lighter or something, you get a flame that goes up. That's because of it's less dense than the air around it, and it gets pushed up. If you do the lighter in space, you just get a sphere of fire. It doesn't flow up, right?
Right.
Our guys are metabolizing on the inside, and you're getting this really fast flow on the outside that shoots media up through the center and completely overcomes diffusion limitations. So even with things that are 1 million cells large, they're bigger than fruit flies, they grow just as fast as if they're small. So, we thought diffusion limitation would be like the big thing holding them back. And how do they solve this? And they get it for free from emergent physics, right?
It's very
so
cool.
We sort of, keep discovering how they co-opt the physics of their environment and things that they get kind of for free, and can put them to novel multicellular purpose.
Do you think that your yeast have evolved anything unique, or has everything you've observed also been observed in nature?
Good question.
Because
I
it is a unique
suspect that it's
scenario,
not
right?
unique.
Like what you're putting it through is
Yeah.
a unique system in of itself. Like there is very specific variables that are being placed on that system. So it wouldn't be impossible with 9,000 generations that you have evolved something that nature hasn't, because nature hasn't replicated those system parameters, right?
Yeah, it's very possible. I haven't spent a ton of time thinking about that, to be totally honest. My suspicion is that nothing we have is truly unique, but that we haven't sampled nature very well, and so maybe hard to know, right?
You can't know.
So like entanglement, I was working with, my main physics collaborator, Peter Juncker, on entanglement, and we were like, wow, this is like a general, powerful mechanism multicellular organisms have at their disposal that allows them to form big, tough groups. Where's the prior literature on this? Basically none. And like, what? Like, how is that possible? Well, it's like a few dozen people working on multicellularity total, and no one's worked on entanglement, really, right? And so then we started looking at like other fungal papers where people had done the scanning electron microscopy of mushroom fruiting bodies, and they're all super entangled looking. But has anyone actually quantified if they're entangled? No, right? So it's one of these things where we can't even say like, oh, entanglement is super common in fungi.
Right.
Even though it really looks like it is. In terms of nutrient flowing, the stuff that we're seeing looks shockingly similar to hyphal fungi.
Very cool. That is very cool. Let's go to mycelium networks, fungal intelligence, this sort of stuff, because we're talking about, these ever more complicated fungal systems and them solving puzzles and problems that are really interesting. You hear about this term a lot, fungal intelligence. Is that an accurate term? Is it a scientific term, or is that pseudoscience?
To be honest, it's not really my field, so I don't know that I can weigh in super heavily. I've never used the term myself. That being said, it sounds like a scientific term to me, in the sense that intelligence is, the capacity for information processing and the ability to act upon it. And I mean, again, I'm not even really an expert in intelligence, so your listeners can correct me if I'm wrong. That's how I think of it. And certainly fungi have a lot of that, right? They have a billion-year evolutionary history of solving spatial problems, where they live in very, very heterogeneous landscapes, and they, unlike animals, can't move around. They have to grow through their environment. So there's a lot of investment in resources to get anywhere. And it's perhaps unsurprising that they have pretty sophisticated mechanisms for making decisions about where to grow based on the needs of all of the cells in the organism. And they seem pretty intelligent by any sort of decision making based on information criterion that one might imagine.
Are you familiar with
that's, I think, generally true.
Yeah. Are you familiar with this? I remember listening to a Joe Rogan podcast where all the best science is dispersed, where someone called Paul Stamets, I'm not sure if you're familiar with Paul Stamets, but he studies mycelial networks. He was talking about a study where fungi were placed on this petri dish, and then there was, I'm going to get some details of this wrong, but the essence will be true, where the Tokyo train line was put down on top, and basically somehow the fungi were able to rearrange the Tokyo train line to make it more efficient, and it actually is. And this was sort of one of those examples of fungi being far more complex than we give it credit for, because it operates in this vast, complicated network that possibly we don't understand. Are you familiar with this study, for one, and how accurate
I
you think?
am?
You are?
I'm familiar with that. I've read the paper.
Amazing. Please tell me about it.
Yeah. So first of all, actually not fungi.
Okay.
That is, the organism there is Physarum polycephalum, which is a acellular slime mold, which is actually very different from fungi.
Okay.
Probably shared a
what are the
common
difference?
ancestor with fungi like over 1 billion years ago.
Okay.
And they have this really interesting ecology, right, where they are these you can have these sort of syncytium, they call them, which is like a giant bag of many, many nuclei. So it's multicellular in a weird way. Like it's one cell because there's one membrane bounding millions of cellular nuclei, but it sort of still has that like multicellular flavor where you've taken like effectively millions of cells, merged them into one rather than merging them and having the cell membranes intact. The cell membranes all sort of like spill together, right? So now it's one giant bag of millions of nuclei. And they have these really cool, they are probably the most animal-like lineage that's not animals, and that they have this cool ecology where they cruise through leaf litter and they eat basically everything in their path. And they actually move, right? So the whole thing sort of moves through moist wood chips and stuff, eating fungi, eating bacteria, shredding them in their path. And
Wow.
they can move like meters per day. They're super cool. And they have these fantastic pipelines where they will do cytoplasmic streaming. So they will actually like set up these networks where if there's like resources around, it takes a while to digest them. They typically feed them oat flakes in the laboratory on petri dishes. They will then like make these pipelines between the resources. Like roughly every 20 or 30 minutes, they'll like reverse the direction of flow, and they're like flowing resources and nuclei between these checkpoints. I think they're so cool.
That is
when I first started my lab, I actually tried a project trying to do directed evolution on them for maze solving behavior.
For
we
which
3D
behavior?
cut like maze solving behavior.
Oh, maze solving behavior. Yeah, I've heard of slime molds and
I wanted them to be able to.
There is some literature with slime molds and
Yeah, well,
solving mazes, right?
Yeah. So there is. It's never evolutionary though. There's maze solving where they they put them in a maze and they like watch them explore around and finally find the shortest path and then make a pipeline between the food at the start and the food at the end. But what I actually wanted to select on them, and it didn't, I could never get the experiment working, but someone else should do this, was actually selecting on mazes with different probabilistic outcomes of the correct answer and involving different bet hedging strategies. So like, would you throw all your resources on just going down the first corridor the fastest because every single outcome is correct and we're just going on speed running it, or make it really low probability where it's best to parallel explore multiple pathways, and then that one is the most reliable one to get through first versus one that has to go down, go back, go down, go back, go down, go back. I thought that'd be super cool. But
that would be.
Yeah, it'd be super cool. But I just had too many mechanistic problems, and I should have collaborated with a slime mold person. Yeah, so I mean, my memory of that paper, and it's been more than 10 years from when I read it, was that it wasn't particularly better than the Tokyo subway. It recapitulated a very similar framework because the heuristic mechanisms of connecting trains is similar between the both. It's shortest path finding. It's like a simple shortest path finding algorithm. Again, I could be wrong, right? It's been that probably came out in like 2010 or something. And so it's been that long since I read the paper. I don't think it's like a replacement for human intelligence and computation. To me, the sort of like, this is a better way of designing trains argument was sort of I didn't buy that.
Bit sensational.
Because, well, I mean, I don't know. I wouldn't impute motives, but like, I don't really. We have many more considerations than simply shortest path. We have topography. We have population centers. We have political considerations with who needs to get the pork, the money in their district. We have tons of considerations that humans have to weigh. And I think those are important considerations. And the slime molds don't care about that.
Yeah. They're
just doing shortest path connecting between bits of food, right? And so it is really cool. I think it's super cool. What to me is amazing is that they convergently come to a similar conclusion to what humans would do. And like that tells you, that's again kind of like these manifolds I was saying before. There's certain convergent things that you see as motifs over and over and over in evolution because that's like the best solution for the local ecology. And therefore evolution has kind of driven things to a similar place. And you can predict that those will occur because they have the highest local payoffs. And so to me, that's a manifestation of convergent evolution. Slime molds and humans solve similar problems of connecting things with an efficient network for the same reasons that you want to make efficient short connections between resource hubs with bigger connections between bigger resource hubs and smaller ones between accessory ones. That's super tight, but I don't think it really needs to go beyond.
How on earth is a slime mold doing that though? Like, does it have any sort of primitive nervous system? How does it sense the environment? How is it remotely even doing these resource pathways and creating all of this? How?
I wish I knew.
Okay.
I think that's a fantastic question.
Okay.
I really don't know. I don't know. They don't have, as far as I know, a primitive nervous system. They don't have cell types in the way that multicellular organisms do. Like they can't really have nerve cells when they don't have cells, right? They're one giant cell, right? There must be essentially information gradients between these resources. Every 20 or 30 minutes, they reverse the flow in the direction in which they were going. And so you could imagine, I don't know, I'm just speculating. You could imagine some heuristic where it's like, if there's a lot of food coming from that, like shut down the side paths that aren't bringing the food and reinforce the one that is. And I mean, some very simple algorithm, which gives you an output that's similar. I wouldn't be surprised if computer scientists or mathematically inclined biologists have done those algorithms. But as far as the actual cell biology and really working it out, I'm not aware of that work. Right. I would love to read it if it exists.
But it's likely some pretty basic rule-based reinforcement. As you said, this is working, do more of this. This isn't working, do less of this.
Yep. Yes. That'd be my guess.
What do you think? Do you buy... because I don't know what's good science and what's bad science and related to, these mycelial networks that you hear of, when people make these massive comparisons with the brain, where you have these incredibly complex interlocking pathways a really large ecosystem of parts. And people say these networks are conscious, like these mycelial networks are conscious, they're intelligent. Like, just kind of riff on that. Like, what do you think about these huge, big mycelial networks that are incredibly complex? Do you think it's any way approximating what the brain does, what consciousness, intelligence?
I don't. I mean, I could be wrong, but I don't think so at all. I guess like one thing here is that network complexity, I'm not sure how meaningful that is, right? Complexity doesn't imply function. It may be necessary for some functions, but those have to be, but like those, something like a brain has an incredibly complex architecture because of billions of generations of selection for that brain specific function in computing information, right? Mycelial networks, I think of them as distributed bodies, right? I think of them less in an information theoretic context and more in these organisms are living in extremely heterogeneous environments, and they have evolved to grow quickly in those environments and, outcompete competitors for resources when they become available. Cooperate with, if you're talking about something which is symbiotic with plants, then, they're cooperating with kind of, food producing, carbon producing plants, or they're degrading dead plant material, right? And they do have very complicated, large networks, but in the same way, but so does any multicellular body, right? If you just look at a body, you have lots of cells that are physically connected to each other. And I could make a network diagram of a tree. I can make a network diagram of a frog. I can make a network diagram of a fungus, and they'll have, I'm sure they'll have different network structures. The fungus may have more modular architecture with like less central control and more sort of like, nodes that flare out kind of thing. I would actually guess that most of these large fungal networks are also highly fragmented. Anything that's been around a long time is probably breaking frequently and having necrotic spots, and is less one giant functional organism and more a population of thousands of separate clonal organisms, right? In order to sort of like argue that they have consciousness and intelligence, well, first of all, you got to define what consciousness is. And I don't know, like now we're getting into probably what the cognitive scientist version of what cell types are is to evolutionary biologists, or species are to evolutionary biologists. Like, oh, we can spend the next 3 hours debating what we mean by the word consciousness. Like, but is that productive? Whenever definitions come up, a friend of mine recently said something which I loved. Definitions cannot be right or wrong. They can only be more or less useful.
Right.
Which I really like, because it's true. Definitions are they are something which is created by humans. They are fundamentally arbitrary.
Yes.
And they can be more or less useful, but let's not like get hung up on what the true meaning of this word is, because that's a logical fallacy.
I agree with that. I think it's the big problem with the free will debate is that the two people can be having a conversation but have no agreement on what they mean by free will. I
felt that way too, which is why I've never cared to have that debate.
Yes. And I agree with the networks and consciousness and intelligence. I was just kind of curious on what your riff would be on that, as someone who has probably spent more time than almost anyone in the world looking at fungal networks. Is there anything else?
I wouldn't say that. There are lots of fantastic mycologists that work with outstandingly complicated and cool fungi in nature, and mine are much more in the lab and different from what someone is studying. Some of these really cool natural organisms may think. So I would certainly withhold a little bit of space for their opinions, because like we may have different opinions on this.
Right. Well, I'll ask you again when you hit 50,000 generations, and then we'll
Cool.
see if your opinion's changed. This has been so much fun, Will.
Excellent.
I need to
likewise.
I need to ask one final question, because you brought up an interesting point, which was, it seems that life is incredibly improbable, but you kind of brought up the idea that, maybe it's actually not too improbable. You have, when all of the components are there, life should emerge, and it's emerged many times.
Well, okay, to be clear on life, it's only emerged once that we know of.
That's where I'm going. So,
okay, cool.
So What do you think that there's life on other planets?
Probably. I don't think there's anything special about Earth. It's a very average planet in many ways. And I don't think that there's anything particularly hard about getting chemistry to become Darwinian.
So that's the probably intuition that is going to be surprising for most people, because it seems insanely improbable to me, although I have no intuition on these things, to get chemistry to become Darwinian. But why do you think we haven't found life then on other planets?
We haven't visited any other planet. Well, because it's very hard to visit other planets. Yes, we visited Venus and we had 20 minutes of visibility. Yeah, we visited Mars, and it's really hard to know if there's fossils of microorganisms and where to look. And, if we could send a bunch of samples back, that would be awesome. And it's non-obvious. You would think, in a place like Earth, it's fairly obvious if it's close. You can look at spectroscopic signatures of, biosignatures. We have tons of oxygen, which is out of equilibrium. Wow, if you have that, you probably have, photosynthetic life or something like it. But it's hard to see. It's super hard to see. And it's hard to be unambiguous from afar.
If you had, if you had to guess now, would you say that there is life on any planets that are relatively close to Earth, but we just don't have the chemical tests to determine that outright?
Oh, wow. If we had something with liquid water, I'd be pretty willing to say yes.
So liquid water is the thing for you that,
well,
if that exists, there's going to be some degree of likelihood.
You have so much chemistry. With liquid water, especially with the capacity to have wet-dry cycles, you have all of this potential for chemical catalysis that just is happening. And that's similar, that can imbue, that kind of can lead to the types of life cycles that I think can drive Darwinian evolution of chemistry. We do have liquid water underneath, kilometers of ice on some moons of some of our big planets. If I had to guess if there's life there or not, I don't know. I truly don't know, right? I don't have a strong opinion.
Yeah.
It's more of a coin toss for me. I don't think it's super improbable.
Interesting.
But I also don't think it's yeah, for sure, right? So one other thing I want to say on the origin of life,
please,
which is, I think we make a category error with taking from the history of life on Earth and overfitting what we learned from that, right? So we know that life arose once and has persisted to the modern era, in the sense that the existence of LUCA, a last universal common ancestor, the fact that all living things kind of coalesce back to a singular point, means that the origin has led many people to argue that the origin of life on Earth was a singularity. And so singularities are interesting, right? In math, people often say there's 3 categories: 0, 1, and infinity, right? Because anything else 0, that makes sense, infinity makes sense, and 1 makes sense because something's really special. And we then thought, oh man, origin of life must have been really special. It only happened once in 4.5 billion years. And that means it's hard, right? But my suspicion as an evolutionary biologist, not an origin of life person, is that this is a result not of the fact that it was difficult to get the process going, but a result of something which I call evolutionary priority effects. That once you already have life rolling, and maybe life arose 50 times independently in different lineages, some of them combined, maybe they didn't, but one of them won out. It was the best, right? It would suppress future innovation into that space and potentially drive everything else to extinction. And I also don't think that we should think of life as necessarily a single lineage which gained, the capacity to evolve and then gained all these things vertically by small serial iteration, which is kind of the way that evolution works now, right? Most transmission is vertical. You make an offspring, there might be some mutations there, small iterations and these discrete lines of descent. In a chemical evolution world, the bounds would be much looser. I could see Darwinian evolution of protein-like compounds and RNA-like compounds occurring totally independently and essentially merging into what became life, right? There's no reason they have to be separate. You could have had 10,000 separate sort of catalytic processes in which you have serial hydration and dehydration processes essentially driving chemical synthesis in little pools, right? Which leads to all sorts of interesting chemistry. And these things could totally interact. There's no reason that it wouldn't work the way that life does now, where you have a discrete genome and you have discrete generations and you pop off little babies that are almost exactly like you. Totally different world, right? But once you have something that has those properties, it's just so good. It's so freaking capable. Any rate constants will be millions of times faster than things that aren't organized by biology. The enzymes will be better. The heredity will be better. The ability to evolve will be so much better that it's just going to explode and take off and be the thing that wins. And once you have that environment, you're never going to get origins of life again unless they're in completely different contexts than where modern life lives, because anything that's in the process of building up polymers is food or disrupted by things that are competing for those elements, right? And so I don't think we should overfit that life just arose once. I don't think we should conclude from that it's hard. The other conclusion from life arising once on Earth is that it happened really early. It happened within a few 100 million years, probably, of Earth becoming habitable. And it was super stable, right? Bombard the crap out of the planet. Doesn't matter, right? Like super stable, tons of ecological change, tons of environmental change. Life's really robust. And so for me, that singular origin early says more that it's easy than that it's.
That was really well said. Really, really well said. I'm going to pin you to a yes or no on if you think we'll find life on other planets in our lifetime.
No.
So you're more
because it's too freaking hard to get to them.
Right. And what would be a chemical test that you could determine it without going to the planet? You said that if it has, liquid water, if it has a particular oxygen environment, but aren't those things also assuming that the life is somewhat similar to the life that we have on Earth? Yeah,
that would be true. I mean, I guess if you see chemistry more broadly, and again, I'm a little bit out of my field here. I'm not someone that works on biosignatures, and there's a whole community of people that work on that. But broadly speaking, if you see chemistry that is either, to our knowledge, unique to life on Earth, like it doesn't happen without a lot of biological synthesis, that could be a biomarker, and you might see that spectroscopically. There's been some arguments that we've seen that within the clouds of Venus, but it's hard because other people can say, oh well, I can imagine some way that you might have an abiotic synthesis of the same thing. If you see a ton of oxygen though, like it doesn't tend to persist very long because it's so reactive, right? And yet we know it's generated by oxygenic photosynthesis, which phototrophy, getting energy from light, it evolved twice. It's actually a very cool example of dual singularities where the two different types of photosynthesis inhabit different niches, which are not probably very mutually invadable. That being said, I don't think it's hard to evolve photosynthesis. Like it's not that hard to be able to resonate light into biological energy. And so and the amount of energy that we get from in light in our biosphere from light dwarfs geological contributions to bioenergy. I mean, it's many orders of magnitude higher than the amount of resources that come from geological sources. And so that's a niche that's just, again, going back to this manifolds idea, that is a niche that's sitting there waiting for something to figure out how to exploit it.
Life should get on with it, figure out how to exploit that niche. They need to...
And exactly. And you can kind of imagine that they would. And so if you see a ton of oxygen, it's really out of equilibrium. It's hard to explain how that would just be sitting in an atmosphere and not reacting away with, what's generating it and why isn't it reacting with the rocks that are there, right? Life is if I saw that, I'd be like, oh man, there's got to be tons. Like it's got to be teeming with phototrophs, right?
Yeah. That was really well said. I love it. Last question. I'm curious how you think the technology will develop in the methodology that you employ. And are there any areas of it that you're a little bit scared about? This idea where you can take the myoglobin of a sperm whale and insert it into yeast. Can you take the venom producing protein of a king cobra and put it into a yeast? and then chuck it into the water supply. And you know what I mean? Like, where are the implications? Like,
I know what you mean.
what are the possible implications of like turning organisms into bioweapons if you can reverse engineer, if you can just take one part of an animal and basically implant it into, you know, a tiny microscopic bacteria and then just infect an area with it and no one would know until people just start dropping dead of unknown cause.
All right, I'll say that I don't worry too much. I mean, that's a great question. I don't worry too much about, this has been like something we've been doing for a long time in the field. And fortunately, a lot of these things like crazy venoms, you just couldn't make, it's not just one gene, it's a whole big synthesis pathway. And it requires,
of course,
lots of like post-translational modification that is hard to do in something as simple as a bacteria or even a yeast. So those are tricky. And, you know, people are trying to make lots of bioproducts in microbes because they're good factories for it. But the things that would freak me out in the synthetic biology space are not making proteins in microbes. It's making things that can transmit from person to person, pathogens. So it would be easier to basically just synthesize a virus, one that we no longer have immunity to, like smallpox, or one that is brand new and we've never had immunity to, right? Like, that's not hard to do. And that's super scary to me. Like, I'm much more terrified by a rampaging fire of a pathogen which spreads through an entire 8 billion person susceptible population until it burns itself out to herd immunity. But with high mortality, that is, to me, that's terrifying. Like, I don't worry about AI. I mean, I worry about designer viruses for sure.
Right. That is a scary one. Artificially designed viruses.
Here's another very sci-fi one that's pretty trippy. So I don't know if you saw this report by the National Academy of Sciences on the risks of mirror life.
Mirror life.
So yeah, so life has the molecules that we have. Going back to LUCA, this is great. I get to connect all these things. We have certain chiral orientation to the different molecules that make up life, right? So we use D, I'm not a chemist, I think that's right-handed, sugars, and L amino acids.
Right.
I could be wrong on that. Again, I'm a little over my skis on that, but I'm pretty sure that's right. This has to do with the handedness of these molecules, right?
Yes.
If you were synthesizing them just straight up synthesis, you'd often get an equal mix of left and right-handed stuff. But like living processes just chose one because the other one doesn't work. It doesn't interact with any of our molecules, right? So just chose one and rolled with it, right? And life all uses the same chirality of molecules, which again is like going back to LUCA, going back to one origin. If you did it again, it might be the opposite, right? Pretty cool. People have recently been designing essentially opposite-handed enzymes which can copy DNA, and they're working on making opposite-handed enzymes which can make proteins. And the result of that would be that you might be able to make essentially a mirror world E. coli or something like it, where everything is reverse-handed.
Yes.
And internally, it's completely fine. It's all internally consistent.
Yes.
But what's really scary about that is it would live in basically a shadow ecosystem that it wouldn't interact with the molecules that are the wrong handedness. It wouldn't see it, right? And to a certain extent, it would just eat tons of food that's spontaneously isomerized to the opposite handedness. So there's scraps from the table that it would have access to. But the kind of terrifying thing is that if you were to get a mirror world E. coli in your gut, you couldn't, it may be something which is largely untreatable until we develop mirror world antibiotics. The immune system, I don't think would work properly on it. Normal antibiotics wouldn't affect it at all. The pathogens, the viruses that normally keep these things in check, which you actually really want for bacteria, because the viruses are the main thing keeping their populations from just exploding like crazy, couldn't affect it. Have no effect at all.
What would be the negative of it if it sits there not interacting with your left-handed body because it's right-handed?
It can still grow and take space. And so if it's growing unchecked in your gut or in your bloodstream or in your heart, you could die.
So it's just volume, basically. It's kind of...
Probably, yeah, I guess.
Interesting.
Yeah. So, that's to me, that's like, there should be major controls on any kind of mirror world science experiments. Just don't do it. I mean, also, that's another crazy example that if humans went extinct 10 million years from now and future alien scientists were to visit Earth and take samples, oh yeah, interesting, like there's life here too. And they find these like mirror world ecosystem, they might be able to infer our presence from that, because that's not something that can really happen spontaneously. It takes too many steps to be done by evolution at the same time. Any intermediate that's not fully mirrorized is just completely non-functional. So it can't happen naturally, but it could happen by a intelligent designing entity that has since left the planet, right?
Very cool. That's fascinating. I love the final finish. A good 20 minutes of sci-fi to finish. Sci-fi that you never know when science is going to turn into science fiction, though, with the rate of progress. But that's why I like to touch on it at the end with everyone. Some people bring up, those different examples of where the world is just going to get very strange in the next, who knows, 10, 20, 30 years. So we've left the audience now shivering in their boots about artificially designed viruses and genetic artificial E. coli that their bodies can't interact with. Well, this has been so much fun. I've learned so much. This is definitely not an area of science that I know a lot about, but I certainly know I certainly know more about it now. I am super excited to keep up to touch with your work, how the generations come. If you somehow create some yeast hybrid that's taken over your lab and become conscious and is, taking hostages, that would be a story that I'll look out for.
I mean, yeah, maybe the next interview will be a vaguely wheel-shaped blob of yeast,
right? Pretending to be you, but it's just the
exactly
other side enantiomers.
Totally, it's 10 trillion yeast in a trench coat.
Well, this has been so much fun.
Awesome.
I'll link any part of your work, any of your papers down below, so people can go deep into the research if they're interested, because I think it's so fascinating. This has been so much fun. Thanks so much for your time.
Yeah, thanks for having me on.
I have fantastic news. I know that you love learning about evolution, and I have another evolution podcast to recommend you. It was with Dr. Paul Cizak, and we covered 750 million years of brain evolution. It was one of my favorite episodes to do, and luckily for you, you can watch it right now. All you have to do is click here. Thank you so much. Subscribe to the channel, and I'll see you next time.